For years now, there's been this huge mystery plaguing beekeepers. We've all heard about it, colony collapse disorder. These massive, sudden die-offs with no obvious cause. And, the search for a culprit has been intense. We've looked at everything from pesticides to parasites. But one suspect, a tiny fungus, quickly rose to the top of the list, and it really seemed to hold all the answers. So today, let's dive into that investigation and see what's really going on.
Right? This is the million-dollar question for everyone, from scientists to the folks with a few hives in their backyard. For a while, it seemed like we had a clear answer, but you know what? New evidence is forcing us to take a huge step back and reassess everything we thought we knew.
So, meet our prime suspect, Nosema ceranae. This little guy was originally found in Asiatic bees, but then, in 2006, it was identified in our Western honeybees, and almost immediately, it got linked to colony collapse disorder. The story was that it was this new, super virulent invader. It was the perfect villain for our story. But is that the whole truth?
Okay, to really understand the case against this parasite, we need to kind of go undercover. We need to shrink down and see exactly how this microscopic intruder does its dirty work inside the hive.
So, Nosema disease is, at its core, a gut infection. The parasite basically sets up shop right in the bee's digestive system, and the fallout from that is pretty serious. The infected bee just can't absorb nutrients properly. It gets weaker and weaker, and eventually it can lead to death.
And the way it spreads is just brutally efficient. It all starts when a young bee is cleaning a comb cell that's been contaminated with these tiny spores. Once a bee eats one, the spore acts like this tiny, biological hypodermic needle. It literally injects the parasite into one of the bee's gut cells. From there, the parasite just multiplies like crazy until the cell literally bursts, releasing this flood of new spores to infect more cells and, of course, more bees.
With a life cycle like that, it is really easy to see why Nosema ceranae became public enemy #1. But here is where the case starts to get a little shaky. It all starts with a really basic question. Did we even correctly identify our suspect in the first place?
Okay, so the entire case for ceranae being a whole new species, and not just a variation of its known cousin, Nosema apis, it all hangs on this one number. Scientists found an 8% difference in the DNA of one single tiny gene fragment. And yeah, on the surface, 8% sounds pretty significant. It was enough for them to make the call and say, yep, new species.
But let's put that 8% into perspective, shall we? We now know the parasite's entire genome. Its full genetic blueprint has over 2,600 genes. That one little fragment they tested, it represents less than 0.001% of the total genetic material. Think about that. It's like trying to judge an entire book based on a single, random word. That's a huge problem with that initial classification.
And it's not just us asking this question. Professor Ernesto Guzman, whose fantastic research this explainer is based on, he poses this critical question himself. Is it really scientifically sound to declare a whole new species based on such an infinitesimally small piece of evidence? He argues there just isn't enough data to say for sure that it's not just a different strain of the same old parasite we've known about for years.
So that's the first crack in the case. But the next big hole in this new invader theory is the timeline. The popular story is that this parasite showed up right before colony collapse disorder hit. But you know what? The historical records, they tell a very, very different story.
This revised timeline completely flips the script. Yeah, it was officially identified in 2006 and then linked to CCD in 2007, but we now know it was in North America way, way earlier. We're talking samples from the US in 1995 and even all the way back to 1994 in Canada have since tested positive for Nosema ceranae. So it wasn't a new arrival, it was just newly identified. It had been hiding in plain sight all along.
So you might be asking, well, why the huge delay? Why didn't we find it sooner? And the answer is actually pretty simple: technology. The molecular tools we needed to spot those tiny, subtle DNA differences between N. apis and N. ceranae, they just weren't available back then. We couldn't find it because we didn't have the right kind of flashlight to look for it.
And this really puts the whole origin story in a new light. Professor Guzman offers this fascinating thought experiment. Just think about it. What if that first sample had been identified not in China, but right here in North America, back in Canada in 1994? We'd probably be calling it Nosema albertea today, and assuming it was a homegrown problem all along.
Okay, so it might not be a new species, and it definitely isn't a new arrival. But the last question is really the most important one. Is it actually the super virulent bee killer that it's been made out to be?
Well, when you start looking at the lab results from around the world, the picture gets incredibly murky. It's all over the place.
A research team in Spain, led by Dr. Higas, found that N. ceranae was way deadlier than N. apis. But then, a team in Sweden, under Dr. Fries, ran their tests and found no difference in mortality at all. And then research at the University of Guelph found it only reduced the bee's lifespan by about 14%, which sure isn't great, but it's not nearly enough to explain the catastrophic losses of CCD.
So what's the final verdict here? Well, the evidence is contradictory, it's inconsistent, and it's inconclusive. While Nosema ceranae is definitely a factor in bee health, no one's denying that, the idea that it's the single smoking gun behind colony collapse disorder is standing on some very, very shaky ground.
So if the science is all over the map, where does this leave beekeepers on the ground? It's really easy to feel lost in this whole debate. But the good news is there are some really practical things you can do to protect your hives, no matter which theory turns out to be correct.
And the solution is actually pretty simple. It all comes down to good old-fashioned hive hygiene. We know the main source of infection is those spores hanging out on old soiled combs. So the recommendation is really straightforward. Every year, replace about 1/3 of the old combs in your brood chamber. Just this one action can dramatically cut down the number of spores in the colony, and that reduces the risk of nosema and a bunch of other brood diseases too.
So the case against Nosema ceranae as this lone assassin, it's pretty weak. We know it's been here for decades, its deadliness is totally up for debate, and its very identity is in question. And that leaves us with one final, vital question. If this parasite isn't the primary killer, then what is? The mystery of what's really killing the bees, it continues.